Hey there everybody, this is a video tutorial for grade 12 biology on enzyme regulation, and it fits into the biochemistry strand, usually towards the end of the strand where you have already done biologically important molecules and proteins which make up enzymes, or enzymes are made of proteins, and this is probably about the last topic that you do. The idea for enzyme regulation is that, as you know, enzymes are the workers in the cell or in the body, and they catalyze chemical reactions, whether they're splitting larger molecules into smaller ones in the process of digestion, let's say, or they're putting together molecules, condensing them together to build molecules for storage purposes or to make tissues, that kind of thing. Now, because enzymes are workers, they don't want to work 7 days a week, 24 hours a day. If they were working constantly, or a particular enzyme was working constantly, the product of that enzyme could build up too much. You could deplete substrates, this kind of thing. So they have to be regulated. Now, just looking at the diagram here, you can see that I'm illustrating a substrate like this. All right. So the substrates are labeled here. And a substrate is a molecule that is acted on by a particular enzyme. So in this case, this looks like two, squares joined together. Why don't you imagine that this is maltose, which is a disaccharide made of two glucoses joined together here. So there are two glucoses chemically, covalently bonded together to make this disaccharide, and we need to break down maltose, which you would get in your diet. Let's say you ate some starch, some crackers, potato chips, something like that. Starch is a polysaccharide. It's a polymer of glucose units. And starch is chemically broken down initially in the oral cavity by salivary amylase, and that produces maltose. Now, the enzyme that breaks down the substrate, maltose, the enzyme, which is illustrated right here, this sort of Pac-Man-like structure, the enzyme would be called maltase in this particular case. And you'll note that the enzyme has a space here, and this space is going to fit the substrate. The substrate can fit right into there, sort of like this is a key going into a lock. This, in the old days, was called the lock and key model. It's been revised to now be called the induced fit model, because when the substrate fits into the enzyme kind of changes shape a little bit to hold it better and to strain the substrate so it can do its job. I'm not going to illustrate that here, but you can certainly look into that, but we can still refer to this as the lock and key model. So this is the enzyme, and I'm going to show you some labels right now. This groove or space that I referred to earlier is called the active site. All right, the active site is the site on the enzyme that accepts and fits the substrate, a specific substrate. So if I had a different shape here, it wouldn't fit into that spot. So an enzyme is very specific in the chemical reaction. It catalyzes because of the shape of the active site. It has to fit the substrate. So that's why it's lock and key model. If you had a different key, it may not fit another particular lock. On the enzyme, there may be other sites as well in addition to the active site. One is called the allosteric activation site, and that's a site where an additional molecule, an activator molecule, like I've illustrated here, such as a vitamin or a coenzyme, might fit and do something. We'll talk about that in a bit. And then there's an allosteric inhibition site. So both of these are called allosteric sites. In other words, these are sites other than the active site that can influence the enzyme's activity and regulate it. This one inhibits the enzyme when a molecule fits into here, and I'll show you how that could work as well. So let's take our first example where we have a substrate. So imagine that this stuff is maltose. It's come into the body, and it's now inside a cell. And maltose, the substrate, just collides with the enzyme, and if it happens to hit the active site like a rocket sort of docking in space with another one, okay? If it collides with the active site and it fits, then we now have an enzyme substrate complex, essentially. The substrate is in the active site of the enzyme. Now, the activator comes along, and let's assume this is a vitamin. Now, you probably have taken vitamins in the past, and some of these vitamins, either the ones that you take in a pill-like formula, or they're actually in your food. These vitamins, many of them, interact with enzymes and assist the enzyme in functioning. All right, so in this example, the activator is binding to the allosteric activation site, and watch what it's going to do. All right. I don't know if you can see this. I'm going to move this out of the way. All right, and I'll turn it on, off there. The activator binds, and it does this. It causes a change in the enzyme that assists its function. Now, I've drawn this like little teeth in this Pac-Man-like enzyme so that maybe that helps to grab the enzyme. It's just a graphical way of illustrating that the activator, like a vitamin, assists the enzyme in doing its job. You can also think of it like a battery, all right? Like you've got an electrical device that you have to put a battery in to make it work. So the activator is like the battery, helping the enzyme function. Once the substrate is now bound to the active site of the enzyme will do its job. In this particular case, if this is the disaccharide maltose, the enzyme is going to hydrolyze or split the maltose like this, converting it into products. These products are then released from the enzyme, and you can see that there is still a couple of substrates left. So this next substrate will bind to the active site of the enzyme. The enzyme will do its job, converting substrate into product. You'll note that the enzyme is not changing in the chemical reaction, but the substrate is changing into product. So the enzyme can be reused multiple times. Now, at this point, there is more product than substrate. And these molecules are not guided into the active site of the enzyme. It's just based on concentrations. If you have thousands and thousands of molecules, there's a chance that one of them will bump into that enzyme. There's now more chance that a product will bump into the enzyme and bind to the active site than the substrate. So imagine this particular product comes along and it binds to the active site. Now, it's not going to bind for 24 hours. It might be just a very, very brief thing. But while that product is in the active site, no more substrate has access to it. It's like a car in a parking spot. You can't park your big truck in the same parking spot. So this has stopped the enzyme or regulated the enzyme. This is called competitive inhibition because the product and the substrate are essentially competing for the active site. So this is competitive inhibition. It's also called end product inhibition because it's the product of the enzyme. Here, let me reverse two, all right? There was the substrate. When I converted that substrate into product, it's the product that is competing with the substrate. So it's also called end product inhibition. All right, so that's one way to regulate an enzyme: competitive inhibition, end product inhibition. Another type of competitive inhibition, which is not necessarily a good thing, is with this molecule here. This is a poison. This could be a drug. It could be a toxin, a poison from the environment. And if this enzyme is critical in some kind of chemical reaction to sustain life, imagine a poison coming from maybe a rattlesnake bite or a spider, a poisonous spider. If this poison happens to have a similar shape as the active site of the enzyme, it can fit into that active site. And again, while it is bound to the enzyme, substrate cannot enter the active site. So this is competitive inhibition as well. It's competing with the substrate. And if this substrate has to be converted to products in order for you to live, then that's how this poison could work, by competitively inhibiting the enzyme so substrate cannot get in, and therefore you can't make the products that are necessary for life. All right, so competitive inhibition, allosteric activation, two ways that you can regulate an enzyme. Another type of inhibition is another end product inhibition, but this one's not competitive inhibition. This one is called allosteric inhibition, because the product that was produced when the substrate was broken can come back behind the enzyme and attach to this allosteric inhibitor site, all right, allosteric inhibition site. And when it does that, when the product binds to the allosteric inhibition site, it does this. Watch the active site. See that thing there? It has changed the shape of the active site of the enzyme so that the substrate can no longer fit. Now, the substrate's not competing with the product for the active site. The product actually changed the shape of the enzyme and essentially the active site, so the substrate can't get in. So this is called allosteric inhibition. And in time, as the product is depleted, as there are less of these products, there will be less of these guys to block that site, and the site will go back to normal, and the substrate can fit. So it's all based on concentrations. If you have a lot of substrate compared to product, there's more chance the substrate will come into the active site and be converted into product. As the product builds up, the body's kind of like saying, look, I've got a lot of product, I don't need any more, and I'm running out of substrates. How do I shut this thing off so I don't make more products until I really need them? The way that happens is as the product builds up, there is a tendency for that product to block the enzyme at the active site, competitive inhibition, and therefore no more substrate is converted to product for a little while. As that product is depleted, used up, stored in the body, whatever, as that product disappears, it's like the body saying, well, the product is lower now, I need more product. So the product is gone from the active site, more substrate goes in. And again, as product builds up, another way you can shut down the enzyme is have it come back, bind to the allosteric inhibition site, and block the substrate from getting in there. As the product is depleted or used up, there's less product to allosterically inhibit the enzyme, and therefore more substrate can get in. So that's really three ways that you can regulate enzymes. Use activators to turn them on, all right, like vitamins. Competitive inhibition with the product binding to the active site instead of the substrate. And then I threw in the idea of the poison just because that is competitive inhibition as well. And the third one is allosteric inhibition, where the product binds to an inhibitor site, changing the shape of the active site, so we'll no longer accept the substrate. Okay, hope that helps.